Hi everyone, and welcome to our second Cancer Immunotherapy Early Career Researcher event. So we hosted one last year, and so now we're back again for 2022. I'm really excited to present our new round of early career speakers, as well as our two keynotes. My name is Rebecca Northeast, and I'm a product manager here at Proteintech, and I've been the one who has been organising this event. And just going to start with today's agenda. So first we have our keynote, so Dr. Santiago Zelenay from Manchester, the Cancer Research Institute. Followed by that, we have our 4 ECR speakers. So that's Dr. Liao from the University of Michigan, followed by Chapman from the University of Graz in Austria, followed by Humai Nguyen from the Texas Tech University, then Dr. Mary Budemüller from Detroit. And then following our 4 ECR speakers, we will have Dr. Chapman. We're going to have a roughly 5-minute interval between each speaker to allow people to answer questions. If you have any questions, please do feel free to type them in the box, and then we will get going. So just going to introduce our chair for today. So our chair is Dr. Lydia Lee, and she is a senior clinical fellow at the University College London. Previously, she was awarded a Bloodwise funded junior clinical training fellowship under the mentorship of Prof. Kui Yong and also Dr. Martin Puel at UCL. And here they developed an APRIL-based chimeric antigen receptor for multiple myeloma. This work then formed the basis for the APRIL-CAR trials entering phase 1. And since then, she has been awarded a clinical scientist fellowship by the MRC in 2018. And now Lydia and her team are investigating the role of immune dysfunction in disease progression. So I'm just going to hand over to Lydia, who will introduce our keynote, Dr. Santiago Zelenay.
Hello, thank you very much, Rebecca. I'm excited to be invited to even attend this event, let alone chair it, and particularly looking forward to our keynote speakers. And the first one is Dr. Santiago Zelenay. In 2015, Dr. Zelenay joined the CR UK Manchester Institute, where he leads the Cancer Inflammation and Immunity Group. His group focuses on understanding the underlying mechanisms that mediate cancer inhibitory versus tumor-promoting inflammation in order to design new therapies for cancer patients. In 2017, he received the prestigious CR UK Future Leaders in Cancer Research Prize, and are very much looking forward to hearing him speak about manipulating inflammation to raise cancer immunogenicity and the response to immunotherapy.
Thank you, Lydia, for the introduction. Can you see my slides?
Yes, I can.
Great, thank you. Thanks so much again, Lydia, for the kind introduction, and thanks to the organizer for the opportunity to be here to talk about the work of my group. What I'm going to do in the next 25 minutes or so is to tell you about 2 complementary stories which take me from initial discoveries that I made when I was still a postdoc at the Francis Crick Institute with Cayetano Reyes-Sosa, and how we have expanded and amplified those findings, testing the implications and taking them to very close to be testing this in the clinic. So in this audience, I guess I don't have to give this introduction in which I say that actually immunotherapy has really transformed the landscape of cancer treatment. We know that immunotherapies, especially those based on the use of immune checkpoint blockade antibodies, have really provided unquestioned evidence that the immune system can be a major barrier to cancer, and that immune checkpoint blockade can lead to full remissions even in patients with advanced metastatic disease. So in the context of melanoma, for instance, we have seen patients that have durable benefit from following this treatment with complete remissions that can last for many years. But as you can see here, and this is a plot that shows the objective response rate for immune checkpoint blockade based on PD-1 and PD-1 antibodies that have been approved for more than 16 tumor indications. What you can see is that the response rate, and this is just the clinical response rate, varies across tumor types. So a huge challenge in the field at the moment is the fact that many patients don't respond to the treatments, and actually often even these responses are really non-long lasting. Not only that, these therapies are expensive, and many of these patients face toxicity. So a huge unmet need in the field is why is it that is understanding of why these patients do not respond to treatment. So this has led to a huge amount of active research in the clinic and preclinical models, and have led to this notion that patients whose tumors are not inflamed, they don't contain T cells, and the so-called cold tumors are those that tend not to derive benefit from these treatments. And tumors that are infiltrated by T cells, and in particular CD8 T cells, are those that tend to benefit from treatment and experiencing more long-lasting responses. However, this dichotomy is not perfect. Many patients that have T cell infiltrated tumors do not respond to treatment, and actually some patients with cold inflamed tumors do respond to treatment. However, one could argue that actually all tumors are inflamed entities, and in fact, the most common type of inflammation that is found in clinically apparent tumors is one that I will refer as cancer-promoting inflammation, which is classic cancer-related inflammation that is known to be associated with worse patient outcome, tumor progression, tumor spread, and resistance to therapy. And this type of T-cell inflammation, or the so-called cancer inhibitor inflammation, is less commonly present in tumors, or most commonly present in tumors such as melanoma that are known for being immunogenic. So in this framework, the leading questions in my group is, what really triggers the immune response against cancer? And what are the things that prevent the induction of this immune response against tumor cells? How can we distinguish responders from non-responders, leveraging potentially this information that we learn about the inflammatory composition of tumors? And finally, can we therapeutically make an unresponsive tumor respond to immunotherapy? So over the years, and as I said, what our work has highlighted is that very often cancer cells express the cyclooxygenase prostaglandin 2 pathway, and this is strongly associated with whether the tumor will have this type of inflammation or this type of inflammation, depending on the levels of expression of this pathway. And I will show you in the next minute is sort of the stories and the findings that lead us to believe that this is a very promising target to target in the context of immunotherapy. So what is the cyclooxygenase pathway? So the cyclooxygenase enzymes are the enzymes that are key for the synthesis of prostanoids. They metabolize arachidonic acid into very different prostanoids and eicosanoids, among which prostaglandin 2 is one of the best characterized products of cyclooxygenase activity. And prostaglandin 2 itself has very pleiotropic effects among many cells. Many cells express the receptor for prostaglandin 2, among which EP2 and EP4 are the two receptors that mediate the immune modulatory effects of prostaglandin 2. So what is known over the years is that cyclooxygenase 2 specifically is highly upregulated in several types of human cancers. And that includes lung cancer, breast cancer, colorectal cancer, prostate, ovarian, and many others. So this is a pathway that is very commonly upregulated in tumors. And there's also a lot of vast literature that indicates that COX-2, and specifically prostaglandin 2, one of its products, are directly implicated in promoting most cancer hallmarks. So both COX-2 and PC2 activity have been associated with increasing proliferation of tumor cells, resistance to apoptosis, promoted angiogenesis, among other things. But initially, and this is back when I was a postdoc at the Francis Crick Institute, we were wondering what are the properties of cancer cells that make them immunogenic. And through a series of screening, what we found is that in melanoma cells that were taken from an animal in which melanocytes, from an animal model that spontaneously developed melanoma because they express the most common mutation found in cutaneous melanoma, this B600E, what we found is that these tumor cells spontaneously express cyclooxygenase activity, and as I will show you in a second, this is essential for the ability to evade immunity. So if you take these melanoma cells that are syngeneic with C57 black 6 animals and implant it in the flank of an immunocompetent animal, so these are COX-2 expressing cells, they naturally express high levels of cyclooxygenase 2, these cells from progressive tumors. So what we found is that when we CRISPR out cyclooxygenase activity in these melanoma cells, these tumors form, but then spontaneously got rejected. And this wasn't so surprising because I just told you that it's known that the pathway contributes to tumor cell proliferation, angiogenesis, inhibition of apoptosis. The key finding is that when we put those cyclooxygenase deficient cells in animals that lack T cells and B cells, now these tumors grow perfectly fine, indicating that they didn't have an issue with proliferation. They were perfectly able to form highly angiogenic vascularized tumors, but what they used the cyclooxygenase activity for was to evade adaptive immunity, as in these animals, they don't have T cells and they don't have B cells. Associated with this progressive tumor growth or spontaneously regressing tumors, we wanted to understand what's going on early on in these tumors. What is the type of inflammation that we found? And this is where we found quite a drastic effect. So for instance, in tumors formed by cyclooxygenase competent cells, what we found is that the immune signature, the molecular signature of these tumors early on, is one that is classically associated with cancer-promoting inflammation. So these tumors early on have high expression of factors such as IL-6, CXCL1, or IL-1 beta that are classically known to be associated with a bad patient outcome. In contrast, tumors formed by cycloocygenes deficient cells, early on, before they get rejected, what they have is an inflamed signature, but this is a type of inflammation that is associated with cytotoxic immune factors, including the key chemoattractants of T cells, such as CXCL9 and CXCL10. Effectors of cytotoxic immunity, such as perforin and Granzyme, were all elevated in these tumors. So using these models is that Eduardo Benevita, the first postdoc in my group, started asking, so what are really the key events, what are the signaling pathways that lead to this establishment of cancer-inhibiting inflammation, with the idea that in the understanding this could actually lead us to potential therapeutic targets that we can enhance to increase the efficacy of treatment. So what we did in this case is to implant these cycloocygenes deficient tumor cells, and PTGS is the gene that encodes for cycloocygenes, in mice that lack the pathways that are classically associated with induction of immunity against microbial microorganisms. So in this case, we use MyLTA deficient animals, Tris deficient animals, MAPS deficient animals, STING deficient animals, or MAPS deficient in seagulls. And what you can appreciate in this picture is that in all these cases, these animals, equally as the wild-type animals, reject those tumors. So indicating that these pathways are dispensable for the induction of immunity against these COX-deficient melanoma cells. So we were wondering what is really going on in these tumors. This is quite an unexpected finding, that immune pathways that are classically associated with actually tumor control and microbial infections were totally dispensable for the rejection of these tumors. So we decided to look by multicolor flow cytometry, what is the composition of these tumors very early on, what are the immune cells that are infiltrating these tumors. And what we found is that most of the cell populations were not changing drastically when we look in very early on upon implantation in wild-type animals. What we found is that there were two populations that were consistently changing, and this is not just in melanoma cells, but it's also something that we found across colorectal or BRAF cells using COX component on COX deficient cells of those cell lines. What you find is that in tumor formed by cycloocygenes component cells, there's a drastic and fast accumulation of neutrophils. And this is something that you see here in frequency and in the number. And this is something that is completely gone. The accumulation of neutrophils is gone when the tumors are deficient in cycloocygenes expression. So this is something that you see here. And here we have a control in which we have restored COX-2 activity in the cycloocygenes deficient cells, and this restores the tumor growth and also restores the accumulation of the neutrophils. In parallel, what we saw is that while the neutrophils were accumulated in cycloocygenes component tumors, the NK cells were accumulated in the cycloocygenes deficient cells. So there's actually very few NK cells infiltrating the cycloocygenes component tumors, but what you see is a drastic and acute accumulation of NK cells in tumors that are formed by COX deficient cells. And this is again the quantification that is quite significant. And again, this is something that is observed across many different tumor types. We wonder to what extent these populations that are changing early on are actually responsible for the growth profile of these tumors. So while the depletion of the neutrophils didn't change dramatically, so as far as we tested so far, the growth of these tumors, what we found is that depleting the NK cells prevented the rejection of these COX-deficient tumors. So in this plot, what you see, something that I showed you already, the COX component cells grow progressively, the cycloocygenes deficient cells get spontaneously rejected, the cycloocygenes deficient cells grow progressively when they lack adaptive immunity, but these animals still retain NK cells. When we deplete CD8 T cells in wild-type animals injected with cycloocygenes deficient cells, these cells grow progressively, and when we deplete NK cells, the tumor grow progressively. So indicating while the NK cells are known for being able to directly recognize and kill tumor cells, here the NK cells are present and the tumors are still growing progressively, indicating that the NK cells are now required for the CD8 activity that is essential for the rejection of these tumors. So to us, what potentially the NK cells will be doing, we look back in what's going on early on in these tumors upon implantation of the COX component of the COX deficient cells. So again, this is looking at the molecular profile of these tumors, and here you have, again, the tumor formed by cycloocygenes competent cells in presence of NK cells, or the cycloocygenes deficient cells in presence of NK cells. You see this massive increase in cytotoxic immunity when the NK cells are, when the cycloocygenes is absent in the tumor cells. And what you can see here is that now in these cycloocygenes deficient cells, we deplete the NK cells. Now this increase in cytotoxic immunity is severely impaired, indicating that the NK cells are contributing to this shift in inflammation that happens in the COX deficient cells that is associated with hot T-cell inflammation. So the question is this reduction in these factors because of the NK cells are themselves contributing to these factors, or is it that the NK cells are somehow inducing a shift, remodeling in the tumor microenvironment early on in these tumors? So to directly assess that question, what we did is to sort CD45 immune cells from tumors formed by cyclosine deficient cells in wild-type animals very early on upon implantation in presence or absence of NK cells and perform single-cell RNA sequencing. And what you can see here is that this is the clustering of all the clusters identified by single-cell RNA sequencing for CD45 immune cells within COX deficient cells in presence of NK cells. And this is exactly those same animals, same analysis, where we have depleted NK cells with antibodies. And what you can see, is that actually the composition of the tumors is actually quite similar. The only thing that is missing in this clusters here is this cluster here, which are the NK cells that we are actually specifically ablating in these cells. But while the composition of the immune cells is quite similar and it is not changing dramatically, what we found is that if we actually look at the transcription profiles that are going within this population, this is what's dramatically changing. So the biological processes based on the transcriptomic and the transcriptome of these tumor infiltrating cells is drastically different depending on whether the NK cells are around or not. So for instance, when we take the tumor-associated macrophages, this cluster here, the purple cells that is found in both tumors, and we compare those infiltrating tumors in which the NK cells are present and those in which the NK cells are absent, what we find is that it's a profound enrichment and interferon gamma response. And this enrichment interferon gamma response is something that is consistently observed across all different types of these abundant myeloid populations that infiltrate in these tumors as well. So these tumor-associated macrophages, cluster 2, the cluster 3 here, monocytes, all of them show an increase in interferon gamma response when the NK cells are around in these tumors. And it's not just the interferon gamma response that is increased. We see across all the curated hallmarks that in tumors that are infiltrating, in the macrophages that are infiltrating tumors in which the NK cells are around, we see an increase in interferon gamma response, an increase in allograft rejection. It looks like the metabolic profile is totally different. So they have an increase in oxidative phosphorylation, while the macrophages that are infiltrating tumors in which the NK cells were actively depleted, they have an increase in signaling that are associated with cancer, with bad outcome in patients and with cancer progression, such as hypoxia, PI3 kinase signaling, etc. And this pattern was consistently observed across all those myeloid cell abundant populations that are infiltrating these tumors very early on. Because all these results that I showed you until now were done with a CRISPR deficient tumor cells, and to understand to what extent this is something that is observed to the parental cells, if we for now render the immune cells insensitive to the effects of PG2, and to specifically identify which are the cell populations that might be responding to sensing prostaglandin 2 and prevent and not responding when the tumor produces PG2 that allows the cells to evade immune recognition. What we did is to resort to animals in which the cytotoxic cells, and for that we use a Grandson B-Cre driver, would lack EP4 and EP2, the two receptors that mediate the immunomodulatory effects of prostaglandin 2. So using these animals in which the cytotoxic cells are insensitive to prostaglandin 2, what we find is that if we now implant the cyclosine competent melanoma cells, this cells that produce abundant production of prostaglandin 2, but now the cytotoxic cells, and in these settings we think it's the NK cells that are insensitive to EP2 and EP4 because these are the ones that express considerably Grandson B-Cre, we find that these tumors are now spontaneously rejected. So this is the parental cells. If the NK cells cannot sense the prostaglandin 2, they get rejected, and what we find is that the shift in inflammation that happens in the COX deficient tumors that I showed you a few slides ago is exactly happening in similar fashion, simply by making the NK cells insensitive to prostaglandin 2. So all of this was done with transplantable cell lines. We wanted to what extent we can find evidence that the cyclosine pathway is somehow contributing to the establishment of different types of inflammation in human settings. So to ask that question, what we did is to look at what is the association between the expression of COX2 and all those inflammatory factors that were associated with COX2 expression in the mouse model, and those factors that were associated with the lack of COX2 expression in the COX deficient tumors. So what you see here is a heat map where it shows the correlation between the transcript for COX2 and the transcript for all these inflammatory factors that we derive from the analysis of the mouse model across different tumor types. And what you can see is that those cancer-promoting factors, they tend to strongly positively correlate with COX2 expression across many tumor types. This is not so surprising because, after all, inflammatory factors are known to drive COX-2 expression, and the activity of many of the products of COX-2, including prostaglandin E2, are known to drive the expression of many of the factors. But what is quite striking and we find quite interesting is that across these tumor types, there's an anti-correlation, a significant anti-correlation between the expression of COX-2 and the expression of these factors that we know that when we see them in patients, this is good news. So there's an anti-correlation between these tumors being hot and having and whether there's a COX-2 expression on those tumors, consistent with what we would have found in the mouse models. But we wanted to understand and find evidence as to what extent we can actually integrate this notion about the inflammatory composition of the tumors and integrate these cancer-promoting and cancer inhibitory factors to see whether this allows anticipating patient outcome. So in order to integrate those two different flavors of inflammation, what we did is devise this COX inflammatory signature, which is a ratio between the expression of those cancer-promoting genes divided by the expression of those cancer inhibitory genes. And when you stratify patients across different tumor types, and this is from the TCGA, what you find is that tumors with high COX inflammatory signature, so they have a higher expression of cancer-promoting genes relative to cancer inhibitory genes, what you find is that all those patients do significantly worse in terms of outcome. And this is not just in univariate analysis. When we adjust this analysis, multivariate analysis, by different clinical parameters, including stage, what we find is that in all cases, the COX inflammatory signature is independently prognostic and is strongly associated with worse outcome in patients. This is looking in overall patient survival. Of course, one of the big questions in the field is, what are the patients that are going to respond to immune checkpoint blockade? So to ask to what extent the COX inflammatory signature could potentially help predict and anticipate response to immune checkpoint inhibitors, but we need to resort to clinically available data sets of patient cohorts that were treated with other anti-PD-1, anti-CTLA-4, anti-PD-1 and anti-CTLA-4 combinations, or for instance, in this case, in kidney cancer, anti-PD-1 and anti-VEGF. And in all these independent publicly available data sets, we asked to what extent the expression of the COX-IS is different between patients that benefit or not from those treatments. So these are the non-responders and the responders. And I hope what you can appreciate in this picture is that invariably in all cases, what we see is that patients that did not respond to the treatment were those that had a significantly higher value of the COX inflammatory signature. And interesting, when we benchmark this analysis against classic current immune biomarkers such as PD-1 expression or T-cell inflammation or the infiltration by CD8 T cells, what we find that is the COX inflammatory signature was stronger and more statistically significantly associated with the response in all these data sets. So all this together, this evidence in mouse and in analysis of patient samples indicates that the cyclocycline pathway, it is associated with a shift in inflammation that leads to an accumulation of neutrophils, cancer-promoting factors that are known to be associated with tumor progression, and that when we don't have these, because we render the cells cyclocycline deficient, we have an increase in NK cells, interferon gamma, specific subtype of dendritic cells that are known to be associated with response to immune checkpoint inhibitors and contribute to those responses, and that leads to CD8 mediated response. But of course, the cyclocycline pathway, it is the target of many of those painkillers that we typically take to manage pain or manage inflammation. So we wonder to what extent we can actually therapeutically target the pathway to enhance the response to treatment. So to test that, what we did is to use a preclinical model in which animals bearing a colorectal CT26 cell line in the flank are treated with systemic anti-PD-1 blockade in presence of daily oral doses of celecoxib, a selective COX-2 inhibitor, which in this case we are giving at doses that are considered safe for human use. And what you can see here is a representative experiment. So these are animals that were in this mouse model, and this is quite well known in the field, treated with anti-PD-1. You get about 20–30% of animals that fully respond to the treatment. The celecoxib as a monotherapy didn't use any type of response. But when we use the combination of anti-PD-1 and celecoxib together, we see a synergy where you get about 2/3 of the animals rejecting the tumors. This is something that can be appreciated here, where we have a compilation of different experiments. What we're looking here is at the change in tumor diameter 2 weeks post-treatment, so this is quite early upon treatment. What you can see is that anti-PD-1 at this time point is not inducing significant tumor control. Celecoxib is not, as a monotherapy, also inducing significant tumor control, but in the combination of anti-PD-1 and celecoxib, you get already about 40% of the animals that have fully rejected the tumors. And at the experiment endpoint, what we find is, again, 2/3 of the animals have fully rejected tumors as opposed to only 30% on the anti-PD-1. And this leads to a very significant increase in the survival of these animals. But of course, one of the things we wanted to understand is how is it that celecoxib is enhancing this response? So what we did is to look at the composition of those tumors early following treatment. So this is within 1 week within treatment, when the tumors are still macroscopically similar in size, and look at what is going on in the animals that are treated with anti-PD-1 and celecoxib. And so I was surprised that this is across of different experiments and across different models, but we found that the actual composition of those tumors 1 week following treatment, so when the tumors are still equal in size, was not drastically changing. And actually, many of these populations were not actually changing between the control and treated mice, or the ones that were treated with this combination that induced significant tumor control. But of course, one of the issues when we do these experiments is that, in contrast with the genetic model, the addition of anti-PD-1 or the addition of anti-PD-1 and celecoxib is not inducing control in all animals. So we have a mixed bag of responses in which some animals respond to treatment and some don't. And potentially these dichotomies in the nature of the response to treatment, which is something that is very commonly observed by everyone that is doing this type of experiments in the immunology field, it could have potentially a confounder that doesn't allow us to understand what is going on in the responders, because what we're looking here is the response, a mix of responses and non-responses. So we decided, can we actually exploit that dichotomy in tumor fate to understand the basis for why some animals responding and others are not responding? So in order to exploit this dichotomy, what we did is to devise a transplantable model in which tumors, instead of implanting animals with 1 tumor, were implanted with 2 tumors, contralateral tumors in each flank. And in this case, one of the things that you do, and again, with the same C26 colorectal cancer cells, implanted in both flanks, and if we treat them with anti-PD-1 or anti-PD-1 and celecoxib, the first thing we saw is that the response is concordant, meaning that if 1 tumor in 1 flank responds, the other one doesn't respond. So contralateral tumors that are responsive will be both responding, and if one doesn't respond, the other doesn't respond. So with this concordance that is very high, higher than 90%, as observed by others as well, what we decided to do is, okay, what we can do now is implant both tumors, treat them with anti-PD-1 or anti-PD-1 and celecoxib, and then surgically remove one of the tumors, but infer whether it would be a respondent or non-respondent, depending on the fate of the contralateral tumor. So this is exactly what we did, and this is the growth profile from those experiments. We treated a cohort of animals with anti-PD-1, a cohort of animals with anti-PD-1 and celecoxib with 2 tumors in the flanks, but one of the tumors, 1 week following treatment, was actually surgically removed and analyzed by RNA sequencing. So when we look now at all these cohort of animals in an unbiased hierarchical clustering, what we find is that actually the transcriptional difference between all these different tumors treated with anti-PD-1 or anti-PD-1 and celecoxib can actually cluster in 2 main clusters. And the good news here is that one of these 2 clusters is actually significantly enriched in animals in which the contralateral tumor went on to respond. So that's why we call it the response cluster. In the other cluster here is one in which is significant increase in non-responders, and therefore we can start to see, okay, what are really the difference between the clusters of response and the cluster of mice that responded to treatment and the cluster of animals that did not respond to treatment. And when we do this analysis, what we find is that one of the biological processes that is very strongly enriched in the responder cluster is indeed the interferon gamma response. But here we are looking very early on upon treatment. This is less than a week before, after the treatment with PD-1 or PD-1 and celecoxib. So consistent with what is seen in the clinic and with other studies, what we find is that in the responder animals, very early on, you have what we call an acute interferon response that is associated with the response to treatment. And again, because what our question was, to what extent the inhibition of the COX-2 prostaglandin pathway and how is it that it's promoted and enhanced in the response to PD-1 blockade. We wanted to know what happened when we treat the animals with immunotherapy with the COX inhibitors. So although these treatments do not induce tumor control by themselves, what we found is that monotherapy with treatment acutely and profoundly also leads to an enrichment on this acute response program in animals that are treated with celecoxib. So this is again looking in the tumor molecular profile. Celecoxib by itself drives a drastic shift in inflammation that happens as early as 4 hours following treatment. So if you basically have a tumor-bearing animal, we treat with celecoxib, and now we do this analysis just 4 hours following treatment, what we find is that things like interferon gamma, PD-1, or CXCL10 are statistically significantly induced just following treatment. So overall, we think that we can actually target the type of inflammation we have in tumors using quite well-known drugs such as COX inhibitors, also in collaboration with industry. We have tested the use of EP2 and EP4 antagonists, and we have seen that similarly, both types of drugs can lead to a drastic and fast shift in inflammation that leads to a type of inflammatory profile that in combination with anti-PD-1 induces stronger tumor control because it's associated with factors that increase T-cell infiltration and activity of T-cells. And this has led to the design of 2 trials, and we are not the only ones that are testing this type of combinations in the clinic. It will be openly very imminently here in Manchester at the NHS Christie Foundation Trust, in which we will be basically testing the combination of immune checkpoint inhibitors that are used as standard of care in combination with high-dose aspirin, and this is in this trial in PALA, in which we will be taking samples before and after treatment with PD-1 with or without high-dose aspirin to see whether this induces a shift in inflammation in patients with triple-negative breast cancer. And in LION, this is a basket trial in which we will be testing the standard of care immune checkpoint inhibition in combination with a selective COX-2 inhibitor to see whether this enhances the efficacy of the treatment. And here the chief clinician is Anne Armstrong, a breast oncologist for the Christie NHS Foundation Trust. So as a summary of my talk, I showed you the cancer cell intrinsic inflammatory mediators are essential for elevation of tumor immunity, that NK cells can drive the broad inflammatory remodeling that is characteristic of hot T-cell inflamed tumors, that antagonistic inflammatory profiles can be found within many cancer types in humans and have prognostic value, and that a gene signature that captures these antagonistic inflammatory profiles, these pro and anti-tuberogenic factors, can predict response to immunotherapy, and that the COX-2 prostaglandin 2 EP2 EP4 axis is an independent immune checkpoint which can be pharmacologically targeted to enhance the efficacy of immunotherapy. And with that, I would like to finish by acknowledging many of the collaborators that have contributed to this, but mainly all of these people in my team. And I have actually described the work of Eduardo Bonavita, who has now his group in Milan, the work of Christian Brownlee, Agrin Moeni, and Victoria Pelli, and many of the others have actually also contributed to this work. And of course, I would like to finish by thanking all my funders, specifically Cancer Research UK, which co-funds my institute at the CUK Manchester Institute. And with that, I thank you for your attention, and I would be very happy to take questions.
Thank you very much for that, Santiago. That was a brilliant talk. We're starting to have questions in, and we can encourage everybody to post their questions as they do come along. The first one was from actually Hong Mi, who says, "Thank you for your presentation. Do you observe the inflammatory response associated with COX-2 similar between subcutaneous and orthotopic tumors?"
Yeah, so that's a good question. So we have done this in the context of orthotopic tumors, in the context of breast tumors, and indeed it's something that we see. And something that, for the sake of time, I didn't have time to show today, but I encourage everyone, if it's interested, to look. It's a Nature Communications paper from last week, when we also see this shift in inflammation when we look at orthotopic tumors, also in the context of chemoimmunotherapy combinations, plus minus COX inhibitors. So the answer is yes. So we see this regard. I mean, so as far as we can tell, we also see when we look in orthotopic sites.
Thank you for that. Next one from Marta Smara. Apologies if I said that wrong. How can you conclude that the effect of the combination of alpha PD-1 antibodies and celecoxib is synergistic and not only additive?
Well, the basis for calling this synergistic is because we don't see that the celecoxib monotherapy by itself induced tumor control. So it's actually, we've seen one example of tumor control with monotherapy with celecoxib across many different experiments. So it's really not additive because we don't see any benefit of the monotherapy in our hands. So it's by definition is synergistic. It's PD-1 does a little bit, celecoxib nothing, together do much more. That's why I'm calling it synergistic.
Okay, and I suppose a question for me, how widespread do you think this additive effect will be beneficial to patients? So do you think there'll be a subset of patients who don't benefit from COX-2 inhibition? So
that's a question we still don't know. I mean, we are actively trying to understand, okay, what would be the type of patients that would benefit from this? So in our study last year, and this is also because in the interest of time, I didn't show it, we do find that the patients that tend to show this shift in inflammation when we inhibited the pathway are those that have the cyclosidase pathway already there. And this is something that we did in collaboration with Daniela Tomen and Tom Schumacher from the NKI using tumor fragments, resected tumors from patients, where we see that this shift in inflammation towards a type of inflammation that favors T-cell activation, it happens only when we compare tumors that have the cyclosidase pathway active compared to those that don't. Having said that, that doesn't mean that if a tumor doesn't have the pathway active, might not upregulate it in response to treatment. But although the answer is still we don't know exactly, it does look like it might benefit those that have the pathway active to start with. Whether there's other features of those tumors that would make them respond, whether they need to be already T-cell inflamed or not, these are things that we're studying at the moment.
Thank you very much. I suppose the other thing is also, can you see this being important in improving adaptive immunity at all? So anti-tumor immunity.
I mean, the responses that I was showing are definitely T-cell mediated, right? So it's definitely improving T-cell control. But you're right that, for instance, in the effects that we see, we think that actually PC2 is acting across many different tumor cells, and specifically it has effect on NK cells that is preventing the activity of innate immune cells, but because those innate immune cells are essential to orchestrate the T-cell response. So definitely in the context of immunotherapy and in the context of T-cells, we think that inhibiting the pathway makes a lot of sense. Whether in other settings, whether you have other type of effectors, this might also help is something that at the moment I cannot really say. But I suspect based on the broad activity of prostaglandin 2 we see across many cells, that we think this is actually probably going to help when you have immunity by different type of cells.
No, thank you for that. It's certainly food for thought. But yeah, that presentation was great. So thank you very much for that. I wonder. Thank you. I think we've set the standard quite high for the talks from now on, so it's a bit tough following that one. But the next one is by Dr. Peng Liao, and he received a PhD in China, and he's currently a postdoc at Michigan, and he's going to speak to us about T-cell derived interferon gamma in combination with the arachidonic acid induces immunogenic tumor ferroptosis, serving as a mode, of action for CD8 T cells mediated tumor killing.
Hello, everyone. Can you see the slides?
Yes, I can see them.
Yeah, thank you. Yeah, thank you, India, invited me to have this chance to present our recently published paper, and also thanks to the organizer and yeah, here. So I'm Peng Liao. I'm from the Weipingzhou lab in University of Michigan. So today my talk is about the CD8 T cells and the fatty acid alters the tumor ferroptosis and immunity through the ACL4. So I think Dr. Zelenay already introduced the immunotherapy, the advanced and disadvanced. So that's why I think for that part of background, I think I just can skip. And then for my study, and so we focus on the basic research, and how we want to figure out what is the mechanism and during the immunotherapy. And so this is a summary of how cytotoxic T cell to kill the tumor cells. So we're classify, so the T cell, typical CD8 T cell will be primed by the APC, such as dendritic cell or the other macrophage. Then the T cell will secrete the peripherin and granzyme, and the peripherin will make a hole on the tumor cell or target cells, and then granzyme B will release into the target cell to trigger the apoptosis. And by the way, and the CD8 also can secrete the interferon gamma and TNF-alpha to kind of cytokine to induce the target cell, the senescence. And recently, so our group found out So the interferon gamma secreted by CD8 cells and then combined with the ferroptosis inducers such as other three, they can trigger tumor cell ferroptosis. So and another group also identifies the Gram B come from the lymphocyte also can trigger the tumor cells by ferroptosis. So the Gram A will get into the tumor cells to target on the gastamine B and then to trigger the ferroptosis. So that means during the killing of the cytotoxic CD8 T cells, the different forms of cell death play an important role and to have the programmed cell death in the tumor cells. And so we are focusing on the ferroptosis. So what is the ferroptosis? So ferroptosis is a non-apoptotic ion-dependent form of the cell death. So you have this kind of the hallmarks of the pharmacs. Usually, they will have the polyunsaturated fatty acid phospholipids, and then they will have the redox the active ion, and they will lose the defensity of lipid proximity repair. So this is the three basic character of this ferroptosis. And so go back to the ferroptosis. So what is important? Why it is important? So early on, so our Italian colleague found out that in this ideated model. So when we give the anti-PD-1 immune blockade, so you can see here the anti-PD-1 blockade, you can dramatically reduce the tumor signaling. And then he isolated the sample from the tissue, and then you can see when they use the CD45 to divide into the immune cells and the tumor cells. And interestingly, that he found out in the tumor cells. So in the tumor cells, when we give anti-PD-L1 therapy, the tumor cell will have more the lipid ROS, which is a marker of the ferroptosis. And also very interesting during the immunotherapy. So this is a B16 model. So when we implant the tumor cell into the mice, this is control in the black bar. So you can see the tumor grow very fast. So once we combine with the CT-alpha and the PD-L1 therapy, you can see the tumor dramatically shrink. The interesting here, so once we give the ferroptosis inhibitor, lipostatin 1, so the efficacy of the immunotherapy will block by this inhibitor. So that means this ferroptosis are really important during the immunotherapy. But and also more and more the evidence shows ferroptosis indeed happen in the in vivo, so such as acute kidney injury, such as a neuron disease. But what is the natural ferroptosis inducer mechanism? So whether it's really happening in the in vivo. So this is a very complex summary of the ferroptosis process by the mechanism. So you can see here from the top, so they have the cysteine metabolites, then will go through the GPCase4 to block the ferroptosis. And in this part, and usually the cell will have the lipid droplet or from the outside uptake fatty acids, then used by the ACL4 and then integrate into the phospholipid. Then if the balance between the lipid peroxidation imbalance, so the cell will have the ferroptosis. So we're wondering whether we can give the fatty acid to promote the ferroptosis instead of use the chemical reagent such as alpha-3 uracil. So this idea, so based on this idea, so we combine interferon gamma with different type of the fatty acids, then use treated this mouse melanoma tumor cell, it's called YAM5.2. So very interesting. So interferon gamma alone cannot induce tumor cells too much, and also the individual fatty acid also cannot induce the tumor cell death. Were quite interesting. Only the combination, the interferon gamma plus arachidonic acid, a type of polyunsaturated fatty acid, can dramatically induce the cell death. But we also tested this experiment in the other type of tumor cells. So among different type of tumor cells, so we can consistently get the result. Interferon gamma combined with arachidonic acid can dramatically induce the tumor cell death. So we are wondering, so what type of cell death is this combination? So then we do this experiment in this young model. We give this kind of treatment plus different cell death inhibitor. So the first study one is for the ferroptosis, and the next study one for the necrosis, and the CD8 is for apoptosis inhibitor. So among different inhibitor block with this interferon gamma arachidonic treatment, so only first study one can block tumor cell death, which induced by interferon gamma A. It's not for other instead of other inhibitor. So we are thinking this is this combination induced cell death is ferroptosis, and also we can see the lipid dose can reduce by this first study one. Is not for the other type of the cell death inhibitors. Then we are wondering, so what is the mechanism by this interferon gamma A induce the cell death in the tumor cells? So here, so we already know based on the literature, we know this SL4 is a critical enzyme to use the free arachidonic acid getting into the phospholipid. So here we also generated this knockout cells in the YAM 5.2. Clearly, so the other three is a ferroptosis inducer, which is can dramatically induce the tumor cells in the white type cells, but not in the SL4 knockout cells. And I go back to our model. So in the interferon gamma combined A treatment, so SL4 in the SL4 knockout cells, so dramatically resistant into to response interferon gamma A induced cell death. Also correlated with the lipid loss. You can see it very significantly reduced the lipid loss in the SL4 knockout cells. So here, so we are thinking the SL4 maybe is also the target of the interferon gamma, which is a response for the interferon gamma A induced the tumor cell death. So then we find out in this YAM cells, when we give interferon gamma treatment, interferon gamma indeed can increase the SL4 mRNA level and also protein level. And as we know, the interferon gamma always knows so have the class mechanism goes through the interferon gamma receptor, then trigger the STAT1 phosphorylation, and then have the targeted downstream induced such as alpha-1. So here we also generate STAT1 knockout cell in the YAM 5.2. As here you clearly can see, so once we give interferon gamma treatment in the STAT1 white type cells, the SL4 expression will be induced, but not in the STAT1 knockout cells. And also this mechanism also is can implied into this tumor cell death. So in the STAT1 knockout cells, the interferon gamma and A cannot further to induce the tumor cell death. And so what is the molecular mechanism? When we go to the genomic analysis, we find out on the SL4 promoter region that they have this kind of alpha-1 signaling peak by interferon gamma treatment, but not for the STAT1. So then we use the chip assay to find out indeed the alpha-1 protein can binding on the SL4 promoter region to induce the SL4 expression. So as we know, SL4 is an enzyme to utilize the arachidonic acid, and then we'll change the phospholipid pattern in the entire phospholipid. So then we are wondering during this treatment, interferon gamma and A in the tumor cells. So what is the difference of the different phospholipid change? So here is the heat map. So we use the target lipidomic data analyze to trace. We label the arachidonic acid Then we use the matchback to track the where it go of the A. So very interesting. So we detail analyze the specific the P group and the PC group. As you can see, so the AA is preferred integrate into the carbon 16 and carbon 18 associated phospholipid. So which is ACL4 dependent manner. So ACL4 dependent, you can see in ACL4 knockout cells. So and this type of phospholipid is not didn't increase in the interferon gamma plus AA treatment. So based on this idea, as we know, so the carbon 18, such as the 18 with the one carbon bond, so they have a different form of the fatty acid. So and so here we give another. So all of this polyunsaturated fatty acid, the monounsaturated fatty acid is the same molecule, is carbon 18 plus one carbon bond. So but they just have different the conformation. But very interesting, only this omega-9 oleic acid combined with the interferon gamma can dramatically increase the tumor cell death and also dramatically induce the lipid loss. So they're quite interesting. So I think in the fatty acid combination, so maybe the different form of the fatty acid can synergistically with interferon gamma and A to dramatically induce cell death, which also is ACL4 dependent manner. So we are wondering, so if we go to in vivo data, so how it's going on? So NSG means is an immune defensive mice. So we give the ACL4 white type tumor cells and knockout tumor cells into the NSG mice. So interesting, there is no difference between the white type cell and knockout cells. But in the immune competent mice, the ACL4 knockout tumor is much bigger, much bigger than the white type tumor. And also correlated, we found out the tumor rate and the lipid loss are correlated with the ACL4 knockout tumor. And interesting, we also analyzed the immune phenotype from the tumor sample. As here you can see the CD8 positive cells and the CD4 positive are dramatically reduced in the ACL4 knockout tumor. And also associating the cytokine such as interferon gamma and TNF alpha are significantly reduced in the ACL4 knockout tumor samples. So here, so also we know the interferon gamma are really, we know the arachidonic are really important. So here we test different concentration in the preliminary data. We found out a relatively low arachidonic combined with the PD-L1 can give our synergistic tumor efficacy to block tumor growth. And also we can see in the combination therapy, the interferon gamma TNF alpha can dramatically increase in the CD8 positive cells. And quite interesting. So we knock out the ACL4 in the tumor cells or SAT1, and the AA efficacy to block tumor efficacy is lost in the knockout ACL4 and the SAT1. So here, and I think basically we demonstrate a mechanism, and the arachidonic combined with the interferon gamma can correlatively induce the tumor cell ferroptosis. So which is considered a natural induced mechanism to induce the ferroptosis. And also this manner is depends on the ACL4, and which is goes through interferon gamma and pathway and leave the ACL4 expression. And then and also we found out that ACL4 can mediate the immunogenic tumor ferroptosis. And so maybe A target on ACL4 maybe is a target to synergistic the checkpoint therapy. So finally, I would like to thank the Lab and typical for the VP and the INCA for this guidance, and also we will thank the administrators group to give our very strong support from different sides. Also, we also send those other the lab member to give us this project support. And also we like very important to thank the chemical and all of this targeted lipidomic analysis is supported by this chemical. And without that, I think the lipidomic cannot move forward. And finally, I always thanks again for this Proteintech to organize this very fantastic series related to immunotherapy. And thanks for your attention. I would like to take any question. Thank you.
Thank you, Peng. We've got our first question through the chat, and from Mary Dumula, one of your fellow speakers later on. Did you look at any downstream metabolites of arachidonic acid with combination with interferon gamma, for example, leukotrienes or prostaglandins?
Yes, it's very interesting. So we indeed examined the COX-2 and the PG2 in the microenvironment. So basically, because we used the relatively low concentration of arachidonic acid, so we didn't see the difference between white type cells or white type tumor and knockout tumor. And also during the immunotherapy and the PGI2 didn't change too much. So I think because we didn't use too much arachidonic acid. And another thing, so during the primary data, we found out that when we give different dose of arachidonic acid, the arachidonic maybe play different role. I think when we give a high concentration of arachidonic acid, indeed the arachidonic acid can promote tumor growth. So it's quite an interesting mechanism. Maybe it goes through the COX-2 to prevent the immunotherapy, but we didn't go further in that part.
So you had alluded to the fact that there were therapeutics in development targeting ACSL4, is that right?
Yes.
And would they be agonists?
And no, so far we don't have. So in the in vitro culture, we use some inhibitor, but we don't have agonists. Yeah.
Right. So therapeutically, what's your vision for these findings?
Yeah, I think it just provides a possibility, and I think the ACL4 maybe is a target that can. I think another thing I want to mention. So all of the cell culture is maintaining the 5% FPS. So at the beginning, so when we use 10% FPS to culture tumor cells, and we found out the tumor cell require a high amount of fatty acid, no matter what kind of fatty acid, typical palmitoic acid, oleic acid, arachidonic acid. So the cell maybe already have a lot of, like no matter in the lipid droplet or in the environment. So we're thinking the cell have very high threshold to respond to the fatty acid treatment. So that's why later we change the strategy to go culture the tumor cells in the 5% FPS. And then we found out that in the 5% FPS culture, the tumor cells are sensitive to response to the arachidonic acid and other type of fatty acids. Then also is implied into the in vivo data. And in the 5%, we can observe the ACL4 knockout tumor is much bigger than the white type tumor, but it's not for the 10% culture of the tumor cells. I think there is still a lot of things we don't know. And yeah, I think still have a mechanism we need to find out.
No, absolutely. No, thank you for that.
Yeah.
So do you know of any genetic permutations of ACSL4?
Pardon? So what is?
So are there genetic variations of ACSL4?
This is quite stable. So usually they don't have any too much mutation, deletion. Yeah, when we check by the TCGA. Yeah.
Okay, thank you for that. Thank you very much for that, Peng.
Yeah.
That was very interesting.
Yes, thank you.
We'll go on to our next talk now, which is by Katrin Pansy. Now, Katrin is a PhD student at the Division of Hematology and at the Medical University of Graz. And she's going to give us a talk entitled "The Loss of NR4A1 is Associated."
Hey, this is the Scheiße. So, oops.
Thank you.
Thank you, Lydia, for this kind introduction. My research group is mainly focused on NR4A1 in aggressive lymphomas, and I'm going to present some of my data of my PhD thesis, where we found out that the loss of NF1 leads to a high expression of immune checkpoints. And in co-culture experiment, we showed a reduced survival using marine lymphoma cell lines with NF1 loss in aggressive lymphomas. First I will start with a short introduction in diffuse large B-cell lymphoma, which are the most common subtype of non-Hodgkin lymphomas and affects the B cells. The incidence in Austria is 7 per 100,000 inhabitants. Despite being an aggressive form of malignancy, it is known as a potentially curable form of cancer with a 5-year survival rate of 16 to 70%. The standard therapy right now is R-CHOP. This is a monoclonal antibodies against CD20 called rituximab with 4 known other drugs. In 1/3 of the patients, a relapse is detected after the treatment, or some patients do not respond to the standard treatment, so it's necessary to find another therapy options. As I said, my research group is focused on NF4As. NF4-1, NF4-2, and NF4-3 belong to the NU77 family, and they are working as a transcription factor and are very important in many biological processes like differentiation, proliferation, and apoptosis. Until now, no known endogenous ligand has been identified, but all 3 family members have a similar protein structure with, on the N-terminal side, activation function domain, followed by a DNA binding domain, a potential ligand binding domain, and on the N-terminal side and on the C-terminal side, another activation function domain. In hematological diseases, it is known that the loss of NF1 and NF3 leads to rapid development of acute myeloid leukemia in mice. And my research group found out that NF1 is downregulated in aggressive lymphomas, and an overexpression of NF1 leads to induction of apoptosis in lymphoma cell lines. To functionally investigate NF1, we are working with a lymphoma mouse model, the MMYC mouse. Here is MYC under control of the immune promoter, a promoter of the immunoglobulin heavy chain. This leads to overexpression of MYC in B cells and causes the development of lymphomas with a latency of 12 to 16 weeks. We intercross this MMYC mouse with a NF1 knockout mouse and performed with this cohort a survival analysis and an RNA-seq. The survival analysis showed that mice, or the MMYC mouse with NF1 loss, had a significant faster development of tumors compared to the heterozygous MMYC mouse or the MMYC mouse without NF1 loss. So we also wanted to investigate how NF1, which genes are regulated by NF1, and this is the reason we performed with tumors of the MMYC mice with or without NF1 loss or RNA-seq, and found out that 75 genes are differentially expressed. Genes which are involved in immunoregulation, like DIM3 or BDL2, are significantly upregulated in these mice, which indicates that genes which are involved in immunoregulation and or in immunoregulatory pathways are upregulated by the loss of NF1. A lot of research has been done to investigate the interaction of tumor cells and immune cells. In general, T cells express inhibitory receptors and tumor cells inhibitory ligands and are called immune checkpoints. This interaction inhibits the effective lysis of tumor cells, and therefore the T cells cannot attack the tumor cells anymore. So over the years, 2 inhibitory receptors have been intensively studied, namely CTLA4 and PD1, sorry. And there are corresponding antibodies against these receptors, and these antibodies are already in clinical use. This treatment should enhance the effective anti-tumor immunity by enhancing the cytotoxicity of the CD8 positive T cells and should also enhance the expression of different cytokines like interferon gamma. Further, to find out which immune checkpoints and genes which are involved in the immunoregulation are expressed on our murine lymphoma cells, and because of our RNA-seq data, we performed a gene expression analysis of lymphomas with or without NR4-1 loss. The gene expression analysis showed that mice with NR4-1 loss had a significant lower expression of interferon gamma, but a high expression of the inhibitory receptor PD1 on the corresponding ligands PDL1 and PDL2, as well a higher expression of the inhibitory receptor CDL4 and the corresponding ligands CD8 and CD86. So this indicates that NR4-1 is implicated in the tumor-mediated suppression of immune cells in the mixed lymphomagenesis. Next, we wanted to find out how NR4-1 influences the immune system. So therefore, we performed a transplantation experiment where we injected lymphoma cells with or without NR4-1 loss in immunocompetent and immunodeficient mice, and checked for the tumor development, performed a survival analysis, and another gene expression analysis. In the immunocompetent setting, we found out that mice transplanted with lymphoma cells with NR4-1 loss had a significant faster development of lymphomas and a reduced survival compared to mice which were injected with lymphoma cells without NR4-1 loss. But in the immunodeficient setting, we could not find any difference in the survival. The gene expression analysis showed in the immune competence setting a similar expression pattern compared to the primary tumors. Also in mice transplanted with lymphoma cells with NR4-1 loss had a low expression of interferon gamma, but a high expression of PD1, PD-L1, and PD-L2, as well as CTLA4, CD8, and CD86. But in the immunodeficient setting, we only found a high expression of the inhibitory ligands, but could not detect interferon gamma, PD1, and CTLA4 expression. So this mice lacks on the B, D, and NK cells which we used. So this so it seems that these genes are only expressed on reactive reactive immune cells. So further, we wanted to investigate the immunoregulatory function of NR4-1. And here we wanted to investigate the interaction of the lymphoma cells and tumor-infiltrating immune cells, especially focused on CD8 positive T cells via the immune checkpoints, and it's influenced also by NR4-1. As the previous data demonstrated, lymphoma cells without NR4-1 loss showed a low expression of immune checkpoints, and therefore the T cells should be able to kill the lymphoma cells in a more efficient way compared to the lymphoma cells with NR4-1 loss, which had a high expression of immune checkpoints. So with this co-culture experiment, we wanted to investigate, or we wanted to find out if low NR4-1 expression suppresses the anti-lymphoma immune response via the interaction of these receptors or ligands. For this experiment, we generated murine lymphoma cell lines with or without NR4-1 loss, and then further we isolated CD8 positive T cells from OT1 mouse spleens and activated them for 38 hours. This OT1 T cells possesses a specific T cell receptor which targets ovalbumin. Then my murine lymphoma cells were stained with a vital dye as CFEC low and CFEC high lymphoma cells, and the CFEC high lymphoma cells were then pulsed with overpeptide, and these lymphoma cells, as the CFEC high and low lymphoma cells, were then mixed in a 1-to-1 ratio, and CD8 positive T cells were added in different ratios to induce the T cell specific reaction against the overlabeled lymphoma cells. And afterwards, the lymphoma cell killing was measured via flow cytometry. As you see here in my co-culture experiment, I showed that when we used murine lymphoma cells with NR4-1 loss, we had a reduced lymphoma cell killing compared to the co-culture experiments using lymphoma cells without NR4-1 loss. To further investigate the impact of CTLA-4 on this immune regulatory axis, we also used OT1 T cells with knockout of CTLA-4. As you see here, when you the loss of CTLA-4 leads to a higher lymphoma cell killing. Also in the when I used murine lymphoma cells with NR4-1 loss, so it could somehow enhance the lymphoma cell killing by using this knockout. And the next step was to translate our mouse experiments into our human diffuse large B-cell lymphoma cohort, and divided this cohort in high and low NR4-1 expressing lymphomas, and performed with this gene expression analysis and also checked on protein level. Also on mRNA level, we could show that diffuse large B-cell lymphoma patients with low NR4-1 expression had a significant low expression of interferon gamma, but a high expression of the inhibitory receptors PD-1 and the corresponding ligands, and also high expression of CTLA-4 and inhibitory ligands. On protein level, we could also confirm these findings and found a high PD-1 expression in reactive lymphocytes and a high PD-L1 and PD-L2 expression in NR4-1 low expressing diffuse large B-cell lymphoma patients. So this leads me to the end of the presentation, where we can conclude that the loss of NR4-1 leads to an accelerated MYC-driven lymphomagenesis. The loss of NR4-1 is associated with an upregulation of genes of the PD-1 and CTLA-4 axis. And in our co-culture experiment, we showed that the loss of NR4-1 leads to a reduced lymphoma cell killing. So to sum up, our data define that NR4-1 function as a novel tumor suppressor, which is involved in the lymphomagenesis via the regulation of immune checkpoints. Thank you for your attention. A big thank you to my team, Division of Hematology. Without these guys, this project could not be possible. And also a big thanks to our cooperation partner. Thank you for your attention.
Thank you, Katrin. Until some questions come in, I guess I'll ask some questions of my own. I suppose, is this a known association of, so I suppose you presented DLBCL as very binary in terms of NRFA1 positive or negative or high or low. And is there an association between them and response to immune therapies?
The diffuse large B-cell lymphoma compared, the ICB therapy is not that. No. Don't have that good response rate right now, because diffuse large B-cell lymphoma patients don't have that much high PD-1 expression or PD-L1 or PD-L2 like for the classical Hodgkin lymphoma, which has amplification, for example, for PD-L2 and PD-L1. So, and right now the ICB is not that good. but we also made a gene expression analysis on another immune checkpoint components like TIM3 or HM, and it also shows a significant upregulation of these genes. And maybe it would be better to combine the different types of blockage like PD1 or TIM3, or maybe it's possible to block also with HM, and then you have a much better response rate.
Oh, thank you for that. And Mai has a question. What would be your prediction on the expression of NF4A1 in patients who receive checkpoint blockade inhibitors?
Yeah, as I said, maybe it's better to block with other immune checkpoints, because yeah, how should I say? You have somehow the PD1 expression here, but it also depends on the tumor microenvironment, and we also saw that somehow it's known in the literature that it lacks on T cells in the tumor microenvironment of in diffuse large B-cell lymphomas.
Thank you. I have been told that there are agonists available such as cytosporin B. Do you think they can be therapeutically exploited?
Yes. Because we in our patients, we thought, it's not like in our mouse model where we totally knock out the NF4-1. We detected a weak expression of NF4-1, and maybe this somehow enhanced the NF4-1 expression. And yeah, then we are thinking that also the immune checkpoints, like PD1 or CTLA4 decreases in this case. And so it can be somehow a treatment or to enhance the ICP therapy.
Thank you. And in some of your animal models, I wonder if you could tell us if you did any studies on the T cell phenotype.
Yeah, we also checked on FACS, the amount of T cells. In our MMUC mouse without NF4-1 loss, we had a higher content of T cells compared to our knockout mouse. We also have in our lab B cell specific knockout of NF4-1, and we also detected the similar low expression compared to the MMUC mouse with NF4-1 loss. So it seems that the tumors of these mice have less T cells.
So apart from the actual number, do you think that they had any signatures of exhaustion, for example?
Pardon?
Apart from the actual number, so the phenotype of the T cells themselves? So what I can remember, we also checked our transplanted mouse, so the tumors of the transplanted mice, and we had a higher amount of CD3 T cells and CD3, also the general T cells, a higher number of CD4 positive T cells and CD8 positive T cells. But this also we also then checked on for the PD1 expression, and it seems that they have an exhausted phenotype because they had a higher expression of PD1. And they were somehow exhausted in these mice with NF4-1 loss. Okay, thank you for that. Another question. What are the further steps of your co-culture experiments? For example, would you like to look at CD4s?
Yeah, the next steps are to perform a co-culture with CD4 positive T cells, because in literature we found that they also have some cytotoxicity, and in combination maybe we could also see much more how the whole tumor microenvironment works in this case. So yeah, it's planned in the near future. Then we want to use for this one a combination with the OT1 mice and the OT2 mice, because then they target the MHC2 molecules on our lymphoma cells. And yeah, we also checked that we have some murine, as we self-generated murine lymphoma cells, which also express MHC2 molecules. So yeah, this experiment is also possible in the future.
Thank you. Thank you. Well, thank you very much for that talk, Katrin. And we'll go on to the next talk, which is by Hongwei Nguyen. Now, Hongwei is a PhD student in the lab of Dr. Wood in the Department of Immunotherapeutics and Biotechnology in Abilene in Texas. And her primary research interest encompasses the area of cancer immunotherapy with a focus on listeria-based vaccines and their application as monotherapy or in combination with immunotherapies, and particularly looking at renal cell carcinoma and colorectal. Ah, wonderful.
Can you see my screen? Sorry.
Yes.
Okay. Thank you for the introduction. Hello, everyone. First of all, I would like to say thank you to Proteintech for giving me the opportunity for talking about my work in the lab. So myself is Hongwei Nguyen. I'm a third-year PhD student in the Department of Immunotherapeutics and Biotechnology at Texas Tech University Health Sciences Center. So in the lab, my work is mainly focused on the use of listeria-based vaccine targeting interferon stimulated chain 15 for renal cell carcinoma. So why do we have to develop a new treatment for renal cell carcinoma? Because this is one of the most common cancer type in the United States, which ranks as the 6th and the 8th most common cancer in male versus female, respectively. The 5-year survival rate for renal cell carcinoma patients are pretty low, especially for those who already have metastasis in the distant organs. Their 5-year survival rate is only about 15%. And more importantly, for kidney cancer patients who undergo treatment for a certain period of time, the systemic therapy, which is very effective at the initial stage, will eventually and gradually become intolerable or resistant. So therefore, it is very important to find and develop a new treatment regimens for advanced renal cell carcinoma patients. So among of the therapy that are in development, listeria-based vaccine is one of the most promising one. This is a very popular vector for cancer immunotherapy. So basically, listeria-based vaccines are developed from Listeria monocytogenesis. This is an intracellular bacterial and the most virulent pathogen on Earth. So before turning it into a therapy, most of the virulent genes were deleted to ensure that it is safe to deliver into the patients. And in addition to that, those listeria will be engineered to encode a certain antigen that can enhance the process of antigen presentation and to deliver the antigen to activate anti-tumor immune response. Now we will look closer into the mechanism of action of the vaccine. So after administering into the patients, listeria-based vaccines will preferentially infect those phagocytic cells, such as dendritic cell, by the formation of phagosome. So inside the phagosome, because of the acidic condition, it will trigger listeria to secrete an endotoxin substance called listeriolysin O, which has the ability to break down the phagosome and free the listeria inside the cytoplasm of dendritic cells, and as well as the antigen. Now the antigen will be present on MC class 1 and activate dendritic cell At this stage, dendritic cell will then travel to the regional lymphoid organ and present that antigen to CD8 T cell and activate the antigen-specific CD8 T cell. At this stage, those CD8 T cells have a better ability to recognize the cancer through the ability to, after getting trained from dendritic cell, and then they will travel to the size of the tumor, recognize the cancer cell, and then kill the cancer cell. So from this overall picture, we can easily identify that one of the most important factors for listeria-based vaccine to work is choosing the right antigen. So in our lab, we have different listeria vaccines that target different antigens. And today I'm going to talk about one of the antigen that we are actively pursuing research in that, which is IHC15. So this is in the normal condition when there's a pathogen infection or DNA damage, the cell will upregulate IHC15, which will then translate into a protein inside the cytoplasm. Inside the cytoplasm, IHC15 can either conjugate with other targeted proteins by the process called IHC-inylation, or it can secrete outside the cell as a free cytokine, which will bind to the receptor LFA1 on lymphocytes and trigger the release of interferon gamma and other pro-inflammatory cytokines to fight against those pathogen infections. But interestingly, in the recent years, there's an accumulation of evidence demonstrating the role of IHC15 in cancer progression and development. So to understand the status of IHC15 in different types of cancer, we look into the human data in TCGA, and strikingly, we found that among 24 different cancer types, IHC15 is significantly upregulated in about 17 of them. Especially in renal cell carcinoma, IHC15 is consistently upregulated at transcriptional level as well as translational level. And from the Kaplan-Meier plot over here, we can see that patients who have higher expression of IHC15, they have a bad survival rate. So this indicates that IHC15 can be a good antigen to target to kill cancer. So with that, we hypothesize and investigate if listeria-based vaccine targeting IHC15 will be efficacious against IHC15 expressing kidney tumors in syngeneic mouse models. And so the very first step to check our hypothesis is that we want to confirm the phenomenon that we see in human also represented by our mutant RCC cell lines. So we check the IHC15 expression by qPCR in RANCA, which is a very common cell line used to represent human RCC cancer. And we found that IHC15 in RANCA is much upregulated compared to the normal fibroblast GT3, as well as compared to the normal healthy kidney tissues. So after having this confirmation, we go ahead and design an experiment to test the therapeutic efficacy of our vaccine in syngeneic mouse model. So in brief, we inject 1 million cells of RANCA subcutaneously in Bausi male mice, and then we randomize them to receive either listeria express OVA, which is a non-specific antigen, and the one that express IHC15. We give the vaccine 3 doses, 7 days apart, and we remove the animal at the end of the experiment. From this experiment, we found that our IHC15 vaccine have a good anti-tumor efficacy against syngeneic RCC mouse model. As you can see, the red lab represent tumor growth and burden from IHC15 group, which is significantly different from the one that doesn't have specific antigen, just OVA. And also, when we look into the tumor microenvironment, we found that treatment with our vaccine also enhanced the infiltration of effector T cell, which is demonstrated by the subpopulation of CD8 positive, CD62L low, and also functional T cell, which secrete interferon gamma, and CD8 T cell that positive for both interferon gamma and TNF alpha. So because CD8 T cell is very important for listeria vaccine from the previous results, we want to determine if the efficacy of our vaccine depends on CD8 T cell. And so we perform an experiment to deplete the population of CD8 T cell, and we found that when we neutralize the CD8 T cell in those mice, efficacy of our vaccine is completely abrogated. As you can see, the mice that receive CD8 T cell blockade, they behave exactly like PBS group in terms of tumor burden as well as in terms of survival. So although we have a great effect of our vaccine in renal cell carcinoma, still we don't have complete tumor eradication. So to understand why, we again analyzed the tumor microenvironment, and we found that our vaccine somehow also enhanced the influx of MBSC population, which made the tumor microenvironment becomes immunosuppressive. As well as our treatment also induced PD-1 expression on CD8 T cell that at some point will reduce the effective function of CD8 T cell. So in the future, we might combine our vaccine with therapies that target PD-1 pathways, such as anti-PD-1, or therapies that reduce the MBSC population, such as sunitinib. And we hope that can lead to complete tumor eradication. So to conclude, I have discussed about the role of IHC15 in renal cell carcinoma progression, and we have discussed about the mechanism of action of Listeria vaccine in general, and we see the efficacy of our Listeria vaccine targeting IHC15 in renal cell carcinoma and how it induced PD-1 as well as MBSC population and the direction for the future of my research. Before ending my presentation, I would like to say thank you again for Proteintech for giving me this opportunity, and of course to my mentor, Dr. Wood, my lab members, funding resources, and TTU agency staffs for helping me doing my research. Thank you, and I would like to take any questions.
Thank you for that, Hong Li. Tell me, are there any licensed Listeria vaccines? I mean, this just shows I suppose this shows the lack of my own knowledge about this field.
So thank you for the question. Right now, there's really many Listeria that target different antigens. So one of the most common one is E7, which already in I think phase 2 of clinical trials, and we still have different antigen in the lab, such as CD105. And the other one in the process of developing. So finding a new antigen is important. And I'm sorry, I'm not quite sure that if I can say the name of that antigen yet, because maybe it's related to patent things. But there's multiple antigens that is targeted by Listeria vaccine in this period of time.
Thank you. And renal cell carcinoma and colorectal is what you've listed as your lab interest. Are these particularly high for this antigen?
Yes. So when my PI, Dr. Wood, when he developed the vaccine, he already tested efficacy in breast cancer, which also have higher expression of IHC15, and it correlates with the survival as well. So I'm using the same vaccine, and I'm testing in renal cell carcinoma and colorectal cancer.
Thank you for that. So what's your vision for this? So I suppose where are you taking this next?
I am hoping to do some study to combine with the anti-PD-1 therapy and seeing, first of all, is to see if they synergize with anti-PD-1 to enhance efficacy. And I'm also curious to see how comparative in terms of control the tumor between our vaccine versus anti-PD-1, which is already like a standard of care currently in the clinic for renal cell carcinoma patients.
Thank you. And there's a question here that's how is Listeria-based vaccines administered?
Oh, thank you for the question. Yeah, we are giving the vaccine IP.
Thank you. Thank you very much for that talk. And I think we'll move on to our last early career researcher talk. It is Mary Odemola, who's currently a postdoctoral researcher in Detroit. And her background is that she obtained a PhD at the Berlin Institute of Technology and Sciences in India in 2018. And this is her third year as a postdoc in gynecological oncology at the Henry Ford Health in Detroit, in the lab of Dr. Ramandeep Ratan, where she researches on various dietary interventions and on its effect of epithelial ovarian cancer progression.
Can you see my slides?
Yes.
Okay. Good morning, everyone. And thank you, Proteintech, for giving me this opportunity to share our research in such a platform. And today I'll be talking about targeting the alpha-ketoglutarate dehydrogenase complex, which reduces the immunosuppressive function of CD11BGR1 positive myeloid cells and inhibits ovarian cancer growth. Epithelial ovarian cancer is often termed as whispering disease, also called a silent killer, and it is the leading cause of gynecologic cancer death observed in women, and with the overall 5-year survival rate of 47%. And as majority of the women are getting diagnosed at later stages, the survival rate is dropping to 30 to 35%. Even though immunotherapy has demonstrated significant benefits across various cancers, but disparity in EOC's response to immunotherapy is due to immunosuppressive factors. This led us to think about identifying targetable pathways that are specific to immunosuppressive immune cells, especially myeloid-derived suppressor cells. So these myeloid-derived suppressor cells are expanded in various disease conditions such as tumor microenvironment, and they form an important component of tumor microenvironment that mediate immune suppression. And tumors can recruit these myeloid-derived suppressor cells by producing various chemokines and cytokines. And after recruiting myeloid-derived suppressor cells, these can express inducible nitric oxide, arginase, ROS, and other immunosuppressive cytokines, which can inhibit the function of NK cells, dendritic cells, and T cells. In ovarian cancer, expanded population of MDSCs is associated with poor prognosis and worse survival. And these MDSCs have been shown to promote ovarian cancer stemness, angiogenesis, and metastasis. So what is the role of myeloid-derived suppressor cells in epithelial ovarian cancer? So to begin with, we started profiling the myeloid-derived CD11BGR1 positive population, profiling between various days during the disease progression in different immune organs such as blood, spleen, bone marrow, and tumor. We found that as the disease is progressing, this myeloid-derived population is increasing, and this might be associated with the positive progression of epithelial ovarian cancer. So we next depleted this GR1 positive population using anti-GR1 monoclonal antibody, and we observed that there was a better survival rate followed by decrease in the abdominal circumference as well as ascites volume. On the other side, when we did Ki-67 staining in tumors, we found that the anti-GR1 depletion mice have decreased tumor progression when compared to the control group. And on the other side, we also saw that upon depletion of this GR1 positive cells, we found that there was increase in CD4 and CD8 positive cells. Next, we also did an adaptive transfer of the CD11BGR1 positive cells, where we first injected mice with IDA tumors, and from there we took bone marrow and isolated the GR1 positive cells and adaptively transferred them into tumor-bearing mice and followed them till the end point for the disease progression. So the adaptively transferred GR1 positive group had more disease progress when compared to the control mice, followed by increase in the abdominal circumference as well as ascites volume. We also saw there was increase in the tumor proliferation, which is performed by Ki index. And as I said that these tumor cells recruit the MDSCs by producing various chemokines and cytokines. when we looked at the tumor-produced factors and stromal-produced factors, we found that most of the chemokines and cytokines are significantly increased in the adaptively transferred myeloid cells when compared to the control group. Next, we also demonstrated this using various immunosuppressive markers such as INOS, arginase, STAT3, IL-1 beta, where we found these markers are significantly increased in the ideate tumor GR1 positive cells when compared to the control. Next, we also performed a myeloid-derived suppressor PCR array, and we observed that the tumor-bearing GR1 positive cells are having a lot of chemokines and immunosuppressive markers, which are upregulated when compared to the control mice. And the other part is the activity of immune cells is defined and modulated by energy metabolism and dictated by tumor microenvironment. So we wanted to study the role of metabolic rewiring in regulating the immunosuppressive capability of these myeloid cells. To begin with, we did a CHORS, the bioenergetic pathway analysis, and we found that the ideate tumor GR1 positive cells are depending on oxidative phosphorylation, and this was significantly increased when compared to the control group. Next, we did the targeted metabolomics, and we found that the TCA cycle metabolites and nicotine metabolites are significantly increased in the ideate GR1 positive cells when compared to the control GR1 positive cells. So what was the thing that is fueling the TCA cycle, whether it's glycolysis or whether it's coming from glutamate? So to demonstrate this, we did a fuel flex assay, which shows us whether these myeloid cells are depending on glucose, glutamine, or fatty acid. So when we did this assay, we found that the ideate GR1 positive cells from the tumor-bearing mice are depending more on glutamine when compared to the other two sources, while they also have the capacity to rely on glucose upon inhibition of glutamine. So next, we used different metabolic inhibitors such as 2-DG for glycolysis, BPTS for glutamine, and etamoxid for fatty acid. And we found that BPTS is inhibiting the oxidative phosphorylation of these tumor myeloid cells when compared to the control. So as we know that, as we came to know that this glutamine is playing a major role, next we also used another inhibitor, DON, which is also a glutaminase inhibitor. And we found that this inhibitor is also inhibiting the upregulated oxidative phosphorylation of these myeloid cells. Next, we incubated these myeloid cells in high glucose glutamine condition and low glutamine condition. And we found that this low glutamine condition myeloid cells are having decreased oxidative phosphorylation when compared to the regularly plated glutamine conditions. So to look deeper into the pathway, we did a glucose metabolism array. And this metabolism array contains like different 5 bioenergetic pathways such as glycolysis, TCA cycle, gluconeogenesis, glycogenolysis, and hexospendose pathway. So when we did this array in both tumor-bearing myeloid cells as well as control myeloid cells, we found that the TCA cycle genes and the glycolysis genes are significantly upregulated in tumor-bearing myeloid cells. Among them, DLST is significantly upregulated across like various genes in tumor-bearing GR1 positive cells. And this DLST is a part of alpha ketoglutarate complex. And so further, we validated this using Western, and we found that the ideate GR1 positive cells are having increased DLST expression when compared to the control. So we just want to know what was this DLST coming from, either it's coming from glycolysis source or it is coming from glutamine. So in order to find that, we did again the incubation of these myeloid cells with 2-deoxyglucose and BPTS. And when we done for DLST expression, we found that glycolysis inhibitor is not having any effect on DLST expression, but BPTS is significantly knocking down the DLST expression. Later, we also incubated the cells in regular glutamine conditions as well as low glutamine conditions. And we found that the low glutamine conditions is knocking down the DLST expression. So far, the working hypothesis is that the tumor cells are recruiting various chemokines and cytokines and priming and expanding the myeloid-derived suppressor cells. And these cells are dependent on the oxidative phosphorylation, the TCA cycle, and glutamine is fueling this TCA cycle. And among this, the DLST is significantly upregulated, and it is the part of alpha ketoglutarate dehydrogenase complex, and which is making the tumor more worse. On the other side, these cells are also inhibiting the T cell proliferation. And from the literature, when we were looking at if there are any targets that are inhibiting the DLST expression, I mean, there are no direct inhibitors of DLSD, but there are some drugs that can target this complex. So there is one drug which is like very famous, and it is almost in 25 to 30 clinical trials, and that is CPI-613 or devimistat. So when we looked at which company is working with, and when we found to know that this Raphael pharmaceutical company is working on the CPI-613, and this company has like a lot of clinical trials going on. And when we shared our interest and we talked about our research, they are very happy to provide us the drug. And then we also came to know that this drug is not only acting on alpha-ketoglutarate dehydrogenase complex, but it is also having a dual action. It is also acting on the pyruvate dehydrogenase complex. So next, we started to test the efficacy of this compound in syngeneic mouse model of ovarian cancer. And we took our experimental plan, injected mice with IDH-P53 tumors and divided them into control and CPI-613. And we gave like 2.5 mg per kg, like every 5th day, and followed for survival, tumor burden, and immune profile. To our surprise, we found that the CPI-613 has significantly improved the overall survival rate of IDH tumors, like almost like 23 days, and followed by decrease in the ascites volume as well as abdominal circumference. Next, we also saw that the CPI-613 is suppressing the chemoattractant factors in tumor microenvironment. Next, to look at if this CPI-613 has any effect on the myeloid cells, when we did the immune profiling, we found that the CD11B population is going down upon treatment of CPI-613. Then to our surprise, we also found that the CD11B GR1 immunosuppressive population is also going down upon the CPI-613 treatment. This is further demonstrated by the bar graph. Next, we were looking for the immunosuppressive markers, and we found that the CPI-613 is also decreasing the arginase 1 expression and IL-1 beta expression, which are like immunosuppressive markers for these GR1 cells. Next, we also did a myeloid-derived PCR array to test if the CPI-613 has any other effect on the chemokines and immunosuppressive markers. We found that most of the chemokines and cytokines are decreased upon treatment of CPI-613. And we also found the further validation that we saw that there is decrease in arginase 1 S100A9 when compared to control GR1 cells. Next, we also tested if the CPI-613 has any metabolic effect on GR1 myeloid cells, and we found that this CPI treatment in decreasing the oxidative phosphorylation, which is also further followed by decreasing the DLSD expression. Next, the other mechanism what we found is the CPI-613 is also increasing the M1 by M2 phenotype. In cancer, M1 is known for anti-tumor responses and M2 is known for pro-tumor responses. So upon CPI-613 treatment, we found that the CD38 population, which is known for M1 marker, is significantly increased in the CPI-613, whereas the EGR2, which is a M2 marker, is going down in CPI-613. When we did overall ratio of CD38 by EGR2, we found that the CPI-613 is significantly improving the ratio of M1 by M2 polarization. So I finally summarized that the CPI-613 has like 3 actions. One is like it can decrease the glutamine addiction in the tumor cells and increase the tumor death. And the other thing is it can make the ineffective immunosuppressive myeloid cell phenotype, and it can lead to increased tumor death. And the third mechanism is it can increase the polarization of M1 by M2 cells. I finally thank all my collaborators, especially Raphael Pharmaceuticals, for sharing us the drug, and my mentor for constant support, and my colleagues and all my funding resources. And I'm happy to take any questions. Thank you.
Thank you, Mary, for that lovely talk. I suppose I've got a question. Did you have a chance to have a look at the phenotype of the T cells in the tumor microenvironment?
Actually, we did not look at the specific T cell subset, but when we looked at the overall percentage of the T cells, like we saw a tremendous increase in the CD8 percentage. And we also looked at the interferon gamma percentage, and which is also increased upon treatment of CPI-613.
Thank you. And has there been any work on the perturbation of this particular of the TCA in other cancer types and the effect of MDSC in those?
Yeah, there are some studies like which are always like they are documented before, and in ovarian cancer there are like very few studies going on, and I think ours is the third paper which came out on ovarian cancer and glutamine addiction and myeloid cells.
Thank you. I'm just waiting to see if there's anyone else has any questions that are coming through. No. You were saying that this company has used these drugs in clinical trials. Have they used this particular drug in clinical trials?
Yes, they are, I think, in the phase 3 clinical trial, and they recently started the combinations with immunotherapies. And but these studies are mainly performed in pancreatic cancer and leukemia. So so we started to begin with the EOC, and we are trying our best to start it in clinical trials. So yes.
And is it well tolerated?
Yes. So far, we did not see much toxic things, because like when we did the higher dose responses, we saw the mice were having some problems. But at lower doses, there was no toxic effect, but there was good immune effect.
Right. No, thank you for that. Thank you for that, Mary. That was a lovely talk. And will that come to the thank you. Well, that comes to the end of our full presentations from early career researchers, and we thank you very much for their interesting work and sharing that data with us. Our last speaker is the second keynote speaker. And in my field needs no introduction. It's Dr. Mike Chapman. He trained in medicine in London and did his PhD at Cambridge with Bertie Goetz, as well as Tony Green. And then he went off to the Broad Institute, where he ran a multiple myeloma genomics project, leading to the first high throughput sequencing of this cancer patient cohort. And then having returned to Cambridge, he now has his own lab, focusing on identifying and developing novel cancer immunotherapy targets and on better understanding the biology of cellular therapies. And he does this alongside a clinical practice with a focus on multiple myeloma as well.
Right, is that showing?
Yeah, looks good.
Great, thanks very much. And thank you for the introduction and the invitation to speak here. So I'm going to talk about some of the challenges that drug companies face and that we face as clinicians working with some of these biological immunotherapies. And just to be clear about what we're talking here, there are multiple types of cancer immunotherapy. So we have naked monoclonals, other drugs based on monoclonal antibodies, antibody drug conjugates, and bispecific T-cell engagers. We have chimeric antigen receptor T-cells, which is part of cellular therapies. And then we have checkpoint inhibitors, cancer vaccines, and cytokines. Now, I'm a hematologist. My clinical and research focus are based around hematological malignancies. And so really I'm going to be talking about the first four modalities here. So naked monoclonal antibodies, which engage complement-dependent cytotoxicity and antibody-dependent cellular cytotoxicity, antibody drug conjugates, which internalize toxins into cancer cells, bispecific T-cell engagers, which link a CD3 binding moiety to a cancer protein, and chimeric antigen receptor T-cells, which are engineered T-cells which recognize proteins on the surface of target cells. I will very, very briefly, just one slide, to talk about the challenge of checkpoint inhibition, because it is such an important subject, but it's not an area in which I can claim any expertise. So checkpoint inhibition relies on, as I'm sure all of you know, interrupting the standard, taking the brakes off T cells in host immune system, and they have been absolutely life-changing for some patients with cancer, and have affected cures in situations which were previously thought to be irrevocable. There are some difficulties. Some cancers are more sensitive to others. So non-small cell lung cancer, melanoma, or renal cell cancers are particularly sensitive. Other cancers are more variably targeted. Now, it's probably less due to the cancer type than the actual molecular phenotype of the cells. And so it's a little bit more complicated than thinking about one cancer or another. But even within a sensitive cancer, only some patients respond, and it's not entirely clear why that should be. And these drugs are associated with severe side effects in some patients, particularly once you take the brakes off the immune system. Autoimmunity can be a major problem. However, I've got no expertise in this field, and I'm going to mainly concentrate on this other group of immune therapies. So these all have in common the fact that they target an intact protein on the surface of the cancer cell, whether it's an immune effector cell binding, for example, CD19 or a leukemia cell, or if it's an antibody-based therapy binding some other molecule. So I think one of the major challenges for drug companies going forward, as they try to expand the repertoire of these sorts of therapies, is identifying the target proteins. Now, this is tricky for a few reasons. So RNA and protein correlation expression is very poorly correlated in all cell types, but at the cell surface it is even more. This is even more so the case. So using RNA to predict targets is really very tricky. Of course, you can look at and you can accurately quantitate cell surface proteins using flow cytometry or CyTOF, but you need to know what you're looking at in advance. This is not an unbiased approach. Of course, you can use mass spectrometry, whole cell proteomics, but there are a number of technical limitations. So the problem is that a lot of cell surface proteins are hydrophobic. They're actually not very abundant compared to other proteins, and the quantitation is difficult. So I'm just going to talk through some data that we published, which came out last week in Blood, talking about a way that I hope is may become useful for the field for identifying these cell surface proteins. So we used this technique, plasma membrane profiling, which involves biotin labeling and strep-tavudine pull down of cell surface proteins, of glycosylated cell surface proteins, developed in Paul Lehner's lab at the University of Cambridge. So the proteins are biotin labeled, they're strep-tavudine pull down. There's tandem mass tag labeling and sample pooling, which then allows you to quantitate individual proteins on the cell surface of multiple samples. And we looked at both, my particular interest is in myeloma, so we looked at both myeloma cell lines and primary myeloma samples. This works very well. So we identified over 3,000 proteins, and there's a heavy enrichment for cell surface proteins. So we know that it's pulling down the right proteins. And if you compare the expression as determined by plasma membrane profiling and flow cytometry, there's a very good correlation. So we were confident that we could accurately identify the proteins. However, with over 3,000 proteins, there is another challenge. How does one rank these proteins? How does one select the ideal target? Now, depending on what modality you're interested in, then there's a real balance between whether that target protein, to what extent that target protein is expressed on the tumor, and to what extent that target protein is expressed on healthy tissue, so-called off-tumor expression. So with naked antibodies that have to co-opt the body's own immune system, actually quite high off-target expression can be tolerated with minimal side effects. However, it's absolutely critical that the on-tumor expression is very, very high. So examples of this are CD20 in various non-Hodgkin's lymphoma, CD19 in, well, actually CD20 in non-Hodgkin's lymphoma, and CD38 in myeloma. Also HER2 in breast cancer. As these therapies become more potent as we go down the screen, actually you can have less and less on-target expression, less and less expression on T cell, but then off-tumor expression becomes more important. And so what you're looking for is a cleaner and cleaner target. So when you use cellular therapies and CAR T cell therapies, it's important that your target is really clean and that there's minimal expression outside of the tumor. Another thing that one can also think about is how easy is it to target these proteins, and it's quite complicated to look at epitopes and use epitope prediction, but we used as a very, very simple surrogate. We showed that actually the extracellular domain size is well correlated with the number of epitopes. So this enabled us to look at three things in tandem. So the expression in the tumor cell of interest, so looking across a group of myeloma samples, the inverse of the expression in healthy tissues, and the size of the extracellular domain. And using very simple linear algebra, we can combine these into a vector score. And actually, if one wanted, one could incorporate other information such as essentiality of the protein in CRISPR screens and what have you. And so using this vector score, we can rank proteins. And this is the top 10 list of proteins that we identified in myeloma. And I think it's important to note that 3 of the proteins that we called the most important are all FDA and EMA approved targets in myeloma. So we have CD38, which is a target of a naked antibody therapy. SLAMF7 also targeted a naked antibody therapy, and BCMA, which has been targeted with antibody drug conjugates, bispecific T cell engagers, and CAR T cells. But we were interested to see that actually, despite finding these, we also found some proteins that had either been minimally investigated or not known at all. So we focused on the top ranking protein in our list, this protein SEMA4A. It's a semaphorin, part of the semaphorin plexin system. And this is expressed on normal CD138 cells. So myeloma is it's a cancer of the plasma cells, the immunoglobulin producing plasma cells. And so we do see SEMA4A expression in healthy plasma cells, although it's at a lower level than in malignant CD138 cells. And it's important to note that actually, similar to BCMA, one can actually eliminate a patient's plasma cells with, and this is relatively well tolerated. If we look at other hematological cells down the bottom left, it is expressed elsewhere. This is very common with hematological tumor targets, but it is expressed at greater levels than other bone marrow cells. It's expressed to some extent in monocytes and a little bit in mature neutrophils, but is not expressed in the hematopoietic stem cells. When you look outside the hematopoietic system, using tissue microarrays, the expression is minimal. There's a little bit of expression in lymphoid tissue, and a tiny bit of expression in cerebral cortex. But if one's interested in an antibody therapy, antibodies should not cross the blood-brain barrier, and this should be acceptable for targeting. Another consideration when looking at immunotherapy is the possibility that the cancer cells can downregulate the target protein, and we were interested in whether this would be possible for myeloma cells. So we used short hairpin RNA to knock the protein down, and we see in the top left graphs that actually hairpins that knock down semaphore A result in a loss of growth, suggesting that that semaphore A is essential for myeloma cell growth. If we look in the top right, we can see that knockdown is associated with apoptosis, so it's probably essential for myeloma cell survival. Stromal tissue is often protective against these sorts of phenomena, but we showed that actually whether you have stroma or whether you support the cells with IL-6, loss of semaphore A protein leads to death of myeloma cells. So we thought this made it quite a good target. In terms of the actual moiety, the way that we would target it, we were interested in antibody-drug conjugates. So we looked at whether semaphore A antibody was internalized. So on the left, we see that at time 0, semaphore A is all on the surface, or semaphore A antibody is all on the surface, but at 3 hours it's internalized. It's co-localized with LAMP1, which is a marker of lysozymes, which are important for antibody-drug conjugate activity. And this process takes place within about 2 hours, and it occurs on all cells. So knowing this, we went on and created a couple of different antibody-drug conjugate models. And we showed that, on the bottom left, we showed that these are highly effective in vitro with picomolar sensitivities, that they don't kill cells that don't express semaphore A on the surface. Again, we looked at the effect of stroma, and stroma does not protect the cells from this killing. So finally, we went on and created some xenograft models involving injection of myeloma cell lines into the tail veins of mice. And if you look on the far right, we've got trastuzumab vedotin. Trastuzumab, obviously it's Herceptin, it targets HER2. This is conjugated to the same toxin as our antibody-drug conjugate, but HER2 is not expressed on myeloma cells. And we also use the naked antibody without an antibody-drug conjugate, which is the 5E3 column. So all the mice had established tumors by week 0, but only in the mice treated with the antibody-drug conjugate, the 2 columns on the left, did we see reversal of the tumor. And this led to increased survival of the mice. So in terms of targeting, or in terms of choosing targets, there are still some challenges, but I think this method of plasma membrane profiling, we're not the only group to do this, but we have been one of the more successful groups in identifying, or getting close to identifying, the full surface proteome. It is a good way of detecting cell surface proteins. We introduced this vector-based score, which one can modify as much as one wants, and this is an effective way of ranking targets. but it's clear that actually, I mean, I showed you only a fraction of the actual in vitro and in vivo work that went on, and extensive validation work is still required to get through to having a bona fide target. There are still challenges. In theory, this process could be used for any tumor, but heterogeneous cell populations are somewhat more complex to profile and need a degree of deconvolution after the event, and this is something that we are still working on. So one can look at cell lines, for example, a lung cancer cell line, but to look at primary tissue is of solid organ cancers is challenging. We used whole cell proteomics data sets to look at healthy tissue expression, but as I said, whole cell proteomics is limited for looking at cell surface proteomics, and so what's really needed is a comprehensive cell surface proteomics data set for healthy tissues. And finally, one of the things that one needs to validate any of these targets is a good tool antibody, and the quality of antibodies, as everyone knows, is very variable depending on the target. And particularly for cell surface targets, you need antibodies that target both the whole protein sitting in its native conformation for things like flow cytometry, and you need antibodies that are effective in immunohistochemistry. So I'll finish off talking about some of the challenges of cellular therapies, which is really a current area of research. I'll talk particularly about CAR T cells. A lot of this applies to other cellular therapies such as CAR NK cells, and to some extent about tumor-infiltrating lymphocytes. But CAR T cells are what have really found their way into the clinic, and what have actually been have received marketing authorization and are being used clinically. So a CAR T cell involves it's a chimera between an antibody, so essentially the B cell receptor binding machinery, and the T cell receptor signaling machinery. These are combined in the middle into this chimeric antigen receptor, and these are used to transform patients' own T cells. There are also allogeneic CARs, CAR T cells and CAR NK cells, but these have other challenges. So at the moment, in terms of what's used in the clinic, it's predominantly autologous CARs, so it's the patient's own T cells are transformed with these chimeric antigen receptors. And particularly in hematological malignancy, perhaps less so in solid organ cancer, these have been absolutely transformative. So in chronic lymphocytic leukemia, up top left, we can see shrinkage of lymph nodes. We can see the Kaplan-Meier curves in diffuse large B-cell lymphoma and acute lymphoblastic leukemia. And we can see reversal of PET-avid lesions in myeloma. And it's important to note that all of these patients were at a clinical stage where they'd become refractory to all other known treatments. And so these patients would have succumbed to their disease usually within a matter of weeks. But in some cases, particularly acute lymphoblastic leukemia, diffuse large B-cell lymphoma, we're actually seeing plateaus of the survival curve. So a little bit like checkpoint inhibition, there is a subset of patients who are being cured from otherwise incurable cancers. They're associated with some particular toxicities. There's on-target off-tumor toxicity, which is what I was discussing in the first part of the talk, and this is predominantly this is a consideration in the design phase of the cellular therapy. Then we have cytokine release syndrome, and immunoeffector cell-associated neurotoxicity, or ICANS, and then prolonged cytopenias. I won't really talk about the cytopenias. It's still a matter of controversy as to what actually causes these, but it may simply be that patients have had very, very heavily pre-treated and have bone marrows which are essentially failing. So I'll really focus on these these two middle ones, the cytokine release syndrome and the ICANS. And that's because they are probably the most challenging toxicities that we see in the clinic. So cytokine release syndrome, it's an on-target, on-tumor toxicity. So in other words, it is triggered by the CAR T cell actually doing what it's supposed to do. So as it binds the tumor cell, it leads to a process of a chain reaction of cytokine release. And what we see is this, multi-organ phenomenon that really mimics gram-negative sepsis with profound hypertension, tachycardia, actually renal failure. And this is can often be life-threatening, often requires intensive care therapy to overcome. And although we've got quite good at dealing with this, it's still a major problem and a cause of morbidity and mortality. There was suggestion from, I think, from Nathan Singh's group from the University of Pennsylvania around 2017. They used this sort of in vitro system where you can take a leukemic cell line, you can take engineered CAR T cells, co-culture these, and then co-culture with other cell types. And it turns out that if you co-culture with antigen presenting cells, dendritic cells, macrophages, that's really what drives IL-6 release. And IL-6 is probably one of the predominant cytokines involved in cytokine release syndrome. And this, by putting in a Transwell system, they were able to show that this is not driven by cell-cell contact, but actually driven by crosstalk from cytokines. And then the other, I think, possibly the most interesting in terms of mechanistic, because it's still really quite poorly understood, but in terms of mechanism, this neurotoxicity, this ICANS, is perhaps one of the most interesting toxicities or challenges associated with CAR T cells. And we see a lot of different clinical phenomena associated with this. It usually occurs at a later stage to cytokine release syndrome. Cytokine release syndrome usually occurs within the first few days, but neurotoxicity can occur up to several weeks later. We do see EEG abnormalities, functional MRI abnormalities. A particularly pathognomonic finding is this agraphia, where patients, even though they are able to, even though they maintain a decent mental state, then they're not confused, they just lose the ability to write. It's almost always reversible, although we haven't really been giving CAR T cells long enough to know whether there are any long-term sequelae of this neurotoxicity. It's probably related to neurotoxicity is probably related to cytokine release syndrome, in that careful case series have shown that you always get cytokine release syndrome preceding neurotoxicity if you monitor it carefully enough. The risk factors are similar for both. Findings from post-mortem have suggested that what's happening is that there's breakdown of the blood-brain barrier. This is indicated by this plasmatodendrosis in the astroglial cells. And it's been shown that if you block certain cytokine releases from monocytes, you can prevent neurotoxicity in animal models. So these were a couple of animal models used to investigate cytokine release and neurotoxicity. The top one is from Attilio Bondanza's group, and the bottom one is from Michel Sadelin's group, both published in the same issue of Nature Medicine. In the Bondanza model, the mice are humanized at the fetal livers are injected with human hematopoietic stem cells, which leads to humanization of the immune system, and then you can use ex vivo CAR transduced T cells to treat the mice, and you get a very faithful reproduction of both the efficacy and toxicity of CAR T cell therapy. The Giavidras model from Michel Sutherland's group essentially just involves having a subcutaneous tumor of very high bulk. It's a much less sophisticated model, and it only really replicates the cytokine release syndrome, not the neurotoxicity. What is interesting is that IL-6 seems to be involved in cytokine release syndrome, and blocking it can prevent cytokine release syndrome, but blocking it has no effect on neurotoxicity. However, IL-1 seems to precede IL-6 release, and this can be blocked by anakinra, and so blocking IL-1 will block both cytokine release syndrome and neurotoxicity. So this suggests some sort of hierarchy of cytokine release, and it does talk to the interaction between the two. So how we're looking at cytokine release syndrome and ICANS is we're using patient samples. We're taking samples from patients undergoing CAR T cell for non-Hodgkin's lymphoma and acute lymphoblastic leukemia. We're taking samples pre-harvest for CAR T cells at the time of conditioning chemotherapy, and then for several days after the CAR T cell infusion. We're looking at single cell RNA sequencing of those samples, and then linking the single cell profiles over time to the patient's hospital records. We're interested about whether there are any markers for cellular populations that might be associated with either of these two syndromes. Now, last year, Michael Green's group at the MD Anderson, taking a similar approach. They actually looked particularly or specifically at the cells from the infusion bag, and they found this very small group of ICANS associated cells labeled as IACs on the Tisney plot, and they suggested, and well, they showed that these cells were associated with neurotoxicity particularly. They're a very, very small population. It's not clear what happens to them once they're infused into the patient. And their actual subtype is a little obscure in that they have some features of monocytes and some features of T cells. And what we are interested in now is actually seeing whether we can, remember this is in the infusion bags, this is after patients have been harvested, their T cells have been sent off, they've been transformed. We're interested in whether we can actually detect any markers of these sorts of cells pre-harvest. So in summary, we have these two clinically very prevalent and life-threatening toxicities. So cytokine release syndrome, it's a rare example in clinical medicine of an on-target, on-tumor toxicity. Neurotoxicity is not clear what drives this, and neither of these can be predicted from pre-harvest samples. So it's still difficult to know which patients are going to do well and which are going to do poorly. And I think perhaps, to me, the biggest challenge here is that actually neither of these syndromes were picked up in the first animal models that were performed before the first human trials. So the first time we saw either of these syndromes was in once these drugs went into humans. And so the models that have been developed were retrofitted, if you like, to try to demonstrate these syndromes once we knew they existed. And that's a sort of fairly unusual situation, and it makes prediction for new therapies, it makes them particularly challenging. So just to finish off, put some of our funders there, people who are involved in the plasma membrane profiling work, particularly Georgina Anderson in my lab, and Paul Lehner's lab, who showed us the technique. So thank you for your time. I think, sorry, I'm a few minutes over, but hopefully I can take any questions.
Thank you very much. So the first talk, the first question is coming through the chat. For the CAR T-cell studies were purified, i.e. only CAR-expressing cells and infused. And what do you think the role of other cells in the therapy have in toxicity?
I'm not quite sure. Does that,
Annika, feel free to qualify the question. Do you mean in the manufactured product or in the patient?
So what I will say is certainly what we use now in the in terms of what is infused. If you take the infusion bags are not pure. There are T cells that have not been transformed. There are other cell types in there. So yes, I mean, I'm not sure whether they, to what extent those infused cells are relevant. I suspect it's still the interaction between the cells that pre-exist in the patient pre-infusion and the infused cells. I'm not sure if that answers the question.
Yeah, no, I think. Thank you. And Santiago said, many thanks for your talk. Because of the role of IL-6 and potentially IL-1B in CRS, what do we know about preventing CRS with anti-inflammatories? That's a good question.
Yes, so I mean, we used tocilizumab, which is a IL-6 receptor blocker. We use that clinically. We get through a lot of that. It's a particular requirement that you have to always have a certain supply of it in the hospital before you treat a patient with CAR T cells, and it's very effective at treating cytokine release syndrome. So anakinra, which blocks IL-1, there are some case reports of it working in neurotoxicity. The Norelli paper suggested it worked well to prevent neurotoxicity, but there haven't been any clinical trials to show that it's effective. So actually, we have on a few occasions when we've been really stuck with patients with really intractable neurotoxicity, we've infused anakinra. It has worked on a couple of occasions, it hasn't worked on another couple. What's done clinically is we tend to use corticosteroids, which are quite effective at preventing neurotoxicity. The difficulty is that there have been studies that show that patients who receive corticosteroids do less well in the long term in terms of their in terms of their relapse rates, but it's not clear whether it's because they got the corticosteroids or because they were particularly sick and required the corticosteroids. So I mean that's what's done clinically. I think the other thing to talk about. So Sarah Grushin had an interesting paper a couple of years ago suggesting that you might get lower rates of these events if actually you have less affinity between the CAR and the tumor target, so that the cells bind quickly, but they come off quickly. You get less tonic signaling through the CAR, and that may lead to less toxicity.
How about NSAIDs in particular? I think Dr. Zelenay's got particular interest in those. We don't tend to use them. I mean, not clinically anyway. I've not seen any research on them. I mean, the problem with NSAIDs is that you've one of the reasons we don't like them is because you've already given some myelosuppressive agents. So patients often have cytopenias, and when patients have low platelets, you don't really want to give NSAIDs because of the bleeding risk. So it's not something I've seen done clinically anyway. I'm not sure about animal models. No, certainly. And it's interesting, isn't it? Because CRS must be context and disease dependent as well. So the incidence in myeloma, for example, is far less than in the ALL setting in CD19. So I think there's still a lot to be learned, and then we haven't really got to the point of being able to permutate the inflammasome in a very sophisticated way.
No, I mean, it's anything but sophisticated. I mean, I think it's an interesting field in that, I mean, a very effective field in that it's been predominantly driven by clinical research and trying things out clinically. And the fact is that we're seeing these toxicities in the patients before we try to model them in the lab. And so, you know, I think this has led to quite rapid innovations, but it also means that, yeah, we have a very unsophisticated understanding of the processes involved.
Yeah, no, I would agree with that. Yeah, no, it's quite interesting because Dr. Zelenay was talking in his talk initially about the different aspects of the inflammasome between tumor targeting and tumor suppressing, and the fact that there's nuances within the tumor microenvironment that can be exploited for clinical use. Certainly in the context of CAR T-cell therapy, I don't think we're nearly up to that stage yet at the moment. And we don't really, and at the moment, all our clinical trials, you know, up to 2/3 of patients or more will receive steroids or TOSI in a typical CD19 trial. And we still don't really know how that influences patient outcomes. We don't think it, at the moment, we think overall that improves their outcome because they avoid toxicity, but how that affects CAR efficacy and persistence, for example, is yet to be defined, I think.
Yeah, I think the whole tumor microenvironment area is fascinating because I think where people would, or one of the areas people would like to see the field go is more into the solid tumors, and it's obviously been much less successful there. Part of that obviously is that it's harder to find targets. It's not that these targets are completely clean in the hematological system, but you could obviously you can lose the healthy target cell, you can lose the plasma cell in myeloma, you can lose B cells, and that's well tolerated. Obviously, if your target is on healthy esophagus or healthy bowel or healthy lung, that's a challenge. But it's more than that. It's the fact that the solid organ tumor microenvironment is much more challenging to target for these therapies.
So where do you see CYP4A and sort of myeloma armamentarium?
Well, I'm hoping that we can. So I mean, we're trying to work this up at the drug company at the moment, and I'm hoping that we can get into. I'm hoping we can get into clinical trials, where I suppose it's like all these things. You work through pragmatically, and you start off at phase 1 trials in multiply treated relapsed refractory disease, and I guess dependent on what you see in terms of efficacy and toxicity determines how easy it is to bring those drugs earlier and earlier into the paradigm. I mean, I think one of the nice things about these sort of targeted therapies is that actually working out the interplay between two different therapies, say between a BCMA targeting therapy and a SEMA4A targeting therapy, is that you can actually look at, well, then there will be some BCMA negative cells, are they SEMA4A positive, and vice versa. So I think you can rationally combine therapies a little bit easier with these sorts of targeted immunotherapies.
No, thank you for that. Thank you for another good talk. So I think we've come to the end of our symposium today. I don't know if Rebecca has.
Hi, yeah, thank you so much, Lydia, for being such a great chat, and thank you for all our speakers. I hope everyone's enjoyed it. I just have a couple of company slides to show, and then after that we'll be able to conclude this really wonderful day. So yeah, thank you so much again, and we've had people tune in from all over, so really exciting that we can provide this platform for a global audience and for global speakers as well. So I'm just briefly going to introduce Proteintech as a company in case you're not aware of who we are. So Proteintech, we have our 3 core brands, and through our Proteintech company, we sell antibodies and immunoassays. We actually have antibodies against 13,000 human targets. And through our Chromatech brand, we also sell our nanobody-based reagents. And so these are 15 kilodalton monovalent antibodies that are really excellent for things like immunoprecipitation and immunofluorescence for super resolution microscopy. But for this day, I want to focus on the humankind recombinant proteins. And so we sell cytokines, chemokines, and growth factors, which have been made through our humankind HEC293 cell system. And so because HEC293 cells are a human cell line, this means that they have native human conformation and also native human PTMs. And so this means that these proteins actually have much higher activity than other proteins which have been expressed in, say, E. coli. And a couple of years ago, we were also really thankful to have been certified for our GMP and ISO grade. And this therefore allows our cytokines to be used in cell and gene therapies, such as CAR T cells. So the humankind advantage is that we never use any tags when we're expressing our proteins, and they're also fully endotoxin free and also animal component free. And yeah, just want to say thank you so much to everyone who joined. Really enjoyed listening to all the talks, and thank you to everyone that stayed. We'll be sending out follow-up email recording and also perhaps the slides as well in case anyone missed it. And if you do have any questions or you would like to receive a certificate of attendance for the event, please do email us on the email below. And yeah, once again, thank you so much to everyone who spoke, especially to Lydia, who handled the questions and the speakers so well. So thank you again, and hope everyone has a really good rest of their day. Bye.